Hi, everyone. Welcome. We're really happy to present today and to have Karla, Loss, and Wilson who prepared this lecture for you. As you know, Karla and Wilson have been working really hard to try to develop Stata examples with all the statistical tests and lectures that we are doing in the program. It's a great honor to have them again today. And we're going to give you Karla and so I'm going to stop sharing right now. And Wilson, you're welcome to start. Thank you all.
Thank you very much, Alma. Nice to see you all. I can see some new people here, but as well some people coming from our group, which is great. For those who I haven't met, my name is Wilson Fandino. Let me just start my presentation here. First, share. Okay. So as I said, let me just, yeah. As I said, my name is Wilson Fandino. I'm one of the PPCRT A2 for this course, along with my colleague Karla Loss. And we decided to prepare for you guys this presentation, which hopefully shouldn't take me more than an hour, maybe, or maybe 90 minutes at the most. And I think this is really important and really and a really interesting topic for this week. We are planning to do more meetings as well. For next week, we are already preparing some things for you relating to the topic of meta-analysis and systematic reviews. But for this meeting, we are going to be focusing missing data in clinical trials. First of all, can you hear me well? Just to test.
Yes, we can.
Perfect.
Yeah.
Yes,
perfect.
So I would like to start this presentation by asking you how do you classify the clinical trials? Can anyone give me an idea of how do you classify the clinical trials? Maybe in terms of the design, in terms of the population, in terms of the location of the trial, in terms of many things. Can anyone tell me? By the way, don't hesitate to interrupt me during the presentation, ask me questions to me and to Karla. So we are very happy to solve out your doubts. So can anyone tell me how to classify the clinical trials? Let me see.
What do you mean with classified? How to structure or a study protocol or?
Yes, for example, in terms of the structure, can you give us an idea?
Yeah, at least I can give you an idea regarding the rough structure. It is the abstract, the introduction, and the results section, discussion and conclusions.
Yes.
And then let's say within the study protocol, yeah, it's the introduction, the material and method section, including research question, hypothesis, trial design, recruit strategy, adherence, enrollment of subjects, randomization blinding, eligibility criteria, inclusion, exclusion criteria, adherence strategy, I just said, outcomes, primary, secondary outcome, sample size, statistical analysis, yeah, results and discussion. So roughly spoken.
Great. Thank you very much, Udo. So, so the reason I was asking is because I brought this slide, which might be helpful for you. And in my perspective, this is the main ways to classify the clinical trials. I mean, in terms of the blinding, in terms of the randomization, in terms of the analysis, location, the population, the hypothesis testing, and the temporality of the study. So as you might already know, the blinding, in terms of the blinding, so we can classify the clinical trials into large groups, which are the open label trials and the blinded. Well, it could be single blinded, double blinded, or triple blinded. The randomization we've discussed a few weeks ago. So there are basically four designs in terms of the randomization. So we know that there is a parallel design, the crossover design, the factorial design, adaptive design. In terms of location, so you can actually conduct your clinical trial in a single center or in multiple center, in which case it's a multi-center clinical trial. The population is important as well when you are trying to define a very specific population, and you are going to avoid as much as possible confunders. So we are talking about pragmatic trial. And on the opposite side, when you are more liberal in terms of the population, this is more close to the reality. So this is an explanatory trial. Same for the hypothesis testing. So you know what the superiority trial is. What you know, in a couple of weeks we are going to examine the non-inferiority trials and the equivalence trials, which are not that common, but they might be important in terms of pharmacoeconomics. And in terms of the temporality, so it could be longitudinal trial, for example, a cohort, parallel crossover factorial and cross-sectional trial when you are just doing a clinical trial in a given moment of the time. So, but in terms of the analysis of the trial, we've got two options, which is the intention to treat analysis and the per protocol analysis. So can you tell me what the difference is between the intention to treat analysis, which is here, and the per protocol analysis?
I'm not sure, but from what I understood, intention to treat analysis is that you are going to do an analysis in all the participants that started the study, even the ones that had like a dropout, like you lost follow-up and didn't finish the study. That's what I understand intention to treat is. And maybe, I don't know what's per protocol, sorry.
Excellent, Karen. So thank you very much. And per protocol is exactly the opposite. So intention to treat analysis, I think the key word is the randomization. So when you are analyzing the patients, keeping in mind where the randomization was, and you are accounting for the missing data. So this is an intention to treat analysis. So it doesn't matter what happened to that patient. It doesn't matter if the patient dropped. We have to keep that information in the for the analysis. And on the other side, the per protocol analysis doesn't really bother about the dropouts, and it doesn't really matter where the patient was randomized. So this is why the randomization is so important, because if you don't respect the randomization, so the benefits of the randomization are going to be lost, and then your results might be biased. Okay, but then so you know that you probably know the benefits of the intention to treat analysis. There are a few indications to conduct a per protocol analysis, as you will see in a couple of weeks with the analysis of non-inferiority trials. But generally speaking, the most conservative approach is the intention to treat analysis. And the reason for this is, again, because of the randomization, because the intention to treat analysis respects the randomization. So in that sense, the benefits of randomization are kept, okay? So do you think it's appropriate to plan beforehand this? Do we need to describe in our protocol how it's going to be our analysis, intention to treat or per protocol, or this is something that we decide as we go?
It should be described in the protocol.
Yes, exactly. As I said, most of the clinical trials are intended to be on an intention to treat basis. But what happens if you described your intention to treat analysis in the protocol, but what happens if you've got a lot of missing data, but you don't have plan to deal with? You don't have any plan to deal with the missing data, but however, you are intended to conduct an intention to treat analysis. So basically, you can't really, because there are two main things. I mean, you have to plan your intention to treat analysis, number one, but it's equally important to have a plan for your missing data. Why is it important? Because because there are some strategies to try to prevent the missing data, number one. And number two, because if the missing data is so much, that would be bias your results, as we will see later on. And that will obviously decrease your statistical power. Actually, for the calculation and sample size, don't forget to account for the missing data that you are predicting. And this is usually 20%. So whatever your sample size is, you should increase at least 20% accounted for the missing data. So the missing data is really important because it's very rare to see any clinical trial completely clean, free of missing data. All the patients had the treatment that they were intending of. This is ideal, but this is not usually the case. Okay, so this is just an introduction of why this is important, why it's important to talk about missing data. Okay, so what I like to propose for this meeting is this. So just imagine that the data set is this, and this is what the data set looks like when your data set is complete, when your data set is, when all your patients had the treatment that they were intended of. You didn't have any dropouts. There was good adherence to the treatment. You didn't have any problems with the follow-up. All the patients finished the trial. So this is like the data set, what the data set looks like in an imaginary basis. Okay, so as I say, this is really uncommon to see this data set looking like this, but we are more used to this kind of data set. Okay, so in which case we got missing data. So I would like to start my presentation with a sort of analogy between a patient which is completely fit and well, and another patient which is completely ill, completely ill. So so just as any other patient, we could see the data set as a disease actually, And as the missing data, it could be a disease of the data set, right? So as any disease, so it has prevention strategies, it has some incidents, it has some pathophysiology, some tools to try to diagnose the disease. So this is my proposal for today. Let's imagine that the missing data is just a disease that is affecting our data set. And this is why our data set is looking like this. You got my idea? I hope so. Okay. So, so based on this analogy, we could say that the missing data in clinical research has some sort of prevention strategies. There's incidents which I already spoke about. We are going to talk about pathophysiology of the missing data. We are going to talk about how to diagnose the missing data in your data set, how to treat the missing data, how to follow up once the treatment is done, what would be the prognosis of your data set based on your pathophysiology, and what would be the aftermath or the sequels after having your some missing data in your data set. So what do you think, in generally speaking, is better? Is it better the prevention or the treatment?
The prevention.
The prevention is definitely better than the treatment. So this is the importance of public health, by the way. So, so the first message I would like to leave you is that there are so many ways to try to prevent the missing data. So if you follow these strategies, you probably won't have too many problems about this. But if you don't plan this, you don't have any strategy. So you have to deal with the treatment, which is not always good, as we will see during this presentation. Okay. So let's start with the prevention then. So as I say, there are many ways to do this. I would say in terms of we could classify this in terms of the study design, in terms of the participants, in terms of the investigators, and even after the patient drop the study, we can still do something. So in terms of the study design, for example, it would be a good idea to run a pilot study to test the study protocol and try to identify what the potential difficulties would be for your clinical trial. So you have a rough idea what would be the rate of the missing data for your clinical trial, for example, and how to address these problems, these potential problems. So running a pilot study is not a bad idea to try to prevent missing data and to try to understand your population as well. Okay. The second consideration is to conduct shorter studies when possible with obviously early study endpoints. I mean, obviously, if you're going to conduct a very long study, maybe for 2 years, so you definitely need to expect at some extent missing data. Increasing the sample size, as I explained to you before, according to the rate of dropouts in similar studies, but roughly speaking, in terms of the sample size, as I said, 20% is acceptable to increase the sample size, and more than 20% is a little bit worrying. When you have more than 20% of missing data. But again, it depends on the context of your clinical trial. The next recommendation would be to conduct a withdrawal design. So only the responders to the treatment are randomized after a random period. So in that case, you are going to save some missing data as well, because those who are not responders are not included in the treatment. Avoid outcome measurements with higher probability of missing data. This is obvious for obvious reasons. And in terms of the participants, so obviously try to limit the burden of the participants. For example, try to minimize the face-to-face visits or the number of visits to the hospital, because if in your design, the patient needs to go, I don't know, maybe every single week for the follow-up. So you might expect that sometimes the patient might not be able to go to the follow-up. So this is a missing data, a source of missing data. So that's something to keep in mind as well. There's nothing wrong in providing monetary and non-monetary incentives to the participants to continue in the trial. When possible, obviously, try to low risky medications so patients are not going to drop because of the lack of effectivity of the treatment, whatever the treatment is. And moving to the investigators. So I don't know what happened here. Let me just, yeah. So
I would like to ask,
yes, Karla, please.
to add a great strategy also. Offer the intervention when you offer placebo to the group, the control group, is a great strategy. So even they are in a placebo group, okay, it's blinded, so they will not know. But in the end, if the intervention works, you can say, oh, we will offer you the intervention if it works. So it's also great to keep the adherence of the project.
Yes, good point, Karla, as well. That would be another strategy in terms of the participants. Moving to the investigators, we can do something else about this. For example, enroll investigators adequately qualified to collect data. This is important as well to have people well trained in clinical research, so they can understand what the implications are for them losing too many patients or losing too many follow-ups or having a big amount of missing data. So they should be adequately qualified to collect the data. For example, we usually distribute templates to collect the information, but if the investigator is not being mindful, so it might misspell the information or maybe missing some information that is critical for the trial. So this is why it's so important to train the people who are collecting the information to make sure that they really understand what the implications are for the missing data. And train the investigator to maximize the adherence to the treatment and motivate them to when all these strategies fail. So we can actually, sorry, this is showing that my internet connection is unstable, but I'm hoping you're still.
We have only a quick interruption, Wilson, okay. But now we are hearing you very well.
Great. So even after the dropouts, doesn't really mean, I mean, if the patient drop out, doesn't really mean that he or she doesn't want us to follow up. So we can follow them up even when they decide to withdraw from their clinical trial for whatever reason. And another good idea would be to just try to find out what the reason was for the withdrawal. Ask the patient, why did you decide to withdraw? This is important as well, because it helps us to understand better the patterns of the missing data and understand better, as you will see later on, the what I call the pathophysiology of the missing data. Okay.
Wilson,
yes.
I have a question, a very short question regarding the follow-up of the participants. who dropped out. You written here, follow them up. Wouldn't be, let's say, does it make sense to, let's say, coming back to the prevention, let's say, at the very beginning of the study, that to write down it in the written consent, to agree that with the participants that if the case arises that they will drop out, that the researchers will be allowed, will be entitled to come back and at least to, let's say, have them on call in order to ask some specific questions regarding the needed values, because I can imagine that rather often, let's say, the participants drop out, but they nevertheless are willing to give some information, which cannot be seen only from the medical records. And so far, I think to write it down on the written consent would make sense. Also, let's say, yeah, in order to be clear, to clarify the situation after withdrawal.
Definitely, Udo, definitely. I think it's all about planning, and I don't think there's anything wrong in writing down in the protocol and try to consent the patients that in case that they withdraw, they will give us at least the chance to follow them up just to understand better what happened. I think this is a great strategy as well, and I wouldn't say. I would say most of the patients would agree with this, because I mean, if they decide to withdraw them, so that's their decision. But just to follow them up by a phone call, it shouldn't really bother them. So yeah, this is a great strategy as well. I mean, as much as you can plan before, the better. The better for your prevention strategies. So great. So this is basically kind of framework of what we would do in terms of the prevention of the missing data. And I can't stress enough that this is probably the most important thing, because as I said, if you are having a lot of missing data, you're really in trouble. I mean, imagine that your patients are dropping in a rate of 40% or 50%, something weird is happening there. And that might compromise seriously the feasibility of your clinical trial as well. So this is all about prevention, I think. And this is why I like to make this analogy with the clinical setting in terms of prevention, treatment, diagnosis, and everything. Okay, so let's move on. So we've talked about prevention. I already spoke to you about the incidents and said it is acceptable to have up to 20%, more than 20% you should start to be worried about. And in terms of the incidents as well, I did say to you that it's very rare to see clinical trials reporting no withdrawals, no dropouts, no loss of follow-ups. This is the ideal scenario, but unfortunately it's really, really uncommon. So I would say the chance of the incidence is really high. In the literature, I saw before this presentation that there's something like 95, 97% of the clinical trials usually have, at some extent, missing data. So what do I mean by the pathophysiology? So by now, you should already be a little bit familiar with this, which is missing not at random, missing completely at random, and missing at random. And this is really important as well, because despite the fact that we can assure what the mechanism was for your missing data, where it was missing not at random, or missing completely at random, or missing at random. So we can by no means we can assure that there's no way to see, oh, this is definitely MAR, MCAR, or MNAR. There's no way to do that. But it is important in terms of the concepts, because most of the strategies that I will mention in the treatment of the missing data have the assumption that the mechanism of your missing data was missing at random. Okay, missing not at random is rather unusual, and I will explain to you why in a couple of minutes. Missing completely at random is the worst. Missing completely at random. Sorry, it's the other way around. Missing completely.
It's the opposite.
Yeah.
Yes.
Sorry. Missing completely at random is rather unusual, and this is the ideal. That should be the ideal scenario when your data is not related to the outcome or to the independent variables. Now, missing not at random is the worst scenario because there's no treatment for this. There's no treatment. And missing at random, this is something that we have some strategies to treat your missing data. So as I say, most of the treatments that we have for missing data assume that your missing data was caused by missing at random, okay, rather than missing completely at random, okay? So what's the difference? Can anyone tell me what's the difference between these concepts?
Hi, Wilson. My name is Ana Maria. I think for missing completely at random, there's no influence in observed or unobserved variables or factors. So they say it's actually quite unusual to happen, but it's completely random. There's no external factors influencing the missing data. For missing at random, there's observable factors that influence these missing data, but it's not strictly correlated to the outcome. So for example, if females are less likely to respond a poll or something like that. And not at random, the missingness is due to unobserved factors and it can be related to the outcome. So for example, those with a more severe condition are less likely to attend or respond, and it's related to the measurement of the outcome.
Very well done, Ana Maria. Basically, this is the best definition. This is what we would like to define these concepts here in the PPCR course. So, as Ana Maria said, missing not at random. When your data is missing not at random, that means that there are serious relationship with your outcome, and this is why when there's something related to your outcome, unfortunately, there's nothing to do. There's nothing related, and there's no treatment for this, unfortunately. And your data are going to be definitely biased. So remember that you will always have an outcome, a primary outcome, and you will have some independent variables that explain that outcome. So those independent variables could be divided into two large groups, which are unobserved independent variables and observed independent variables. This is simple. I mean, the observed independent variables are the variables that you decide to measure in your clinical trial. And the unobserved independent variables are those theoretical variables that you missed in your clinical trial. You didn't consider these variables really important to measure in your clinical trial, but unfortunately they were important, and there's no way to retrieve those data because we have no data about those unobserved independent variables. So missing not at random could be either related to the outcome or related to the unobserved independent variable. This is the concept you have to have. And missing completely at random, as Ana Maria rightly said, is something that has nothing to do with the independent variables and nothing to do with the outcome. This is completely at random, so you don't really need to be worried about this. But as I say, this is very uncommon. For example, let's say that you're measuring a blood pressure, for example, in a given hospital, and then the machine that is measuring the blood pressure for some reason was damaged. This is a very simple example. So you will have missing data because for that hospital, because you don't have the machine to measure the blood pressure. But this is not related to the outcome. This is not related to the independent variables, and it's going to be completely random and distributed, therefore distributed evenly in both arms of your study. And lastly, the missing at random is something that by definition can be explained by the observed independent variables. And because they can explain by the observed independent variables, so we can go for those variables and try to adjust your information for those variables. So this is why we can have a treatment for this missing data. Okay, so long story short, missing not at random has no treatment. Missing completely at random doesn't need any treatment, and missing at random is the treatment I'm going to talk about in a couple of minutes. Okay. It's also useful to see, to think about the diagnosis of the missing data in terms of what the distribution is. So when you are finishing the trial, you should look at your data set and try to see whether or not there is a pattern in your missing data that might give you some key information about, oh, this might be because of this reason, or this might be because of these observing independent variables. So that might or might not give you an idea of what happened with your missing data. If you are able to find a pattern in your data set, that might be a really good finding, because you might have an explanation for your missing data. And then this is the treatment that I was talking about. So basically, you again, following the analogy with a disease, we could have a conservative treatment and something that I would like to call an interventional treatment. This is something that you won't find in the literature. It's something that as I say, this is an analogy with a disease, but I think it's a good way to understand and to have everything in your mind regarding the potential options that you have for your missing data. So what do I mean by conservative treatment? This is called a complete case analysis. So that basically means that you are not doing anything, and you are just going to analyze your existing data. You don't care about the missing data, and that's it. So this is a conservative treatment. Is it appropriate? Definitely not. It's not appropriate, but this is the definition of the complete case analysis is this. And this is why I would like to bring this to the discussion. But because we have a number of options for the interventional treatment, the complete case analysis is really inappropriate and is not really recommended in many cases, unless obviously you have a very minor or quantity of missing data, which is again unusual. Okay. So having said that, so the complete case analysis could be divided into available case analysis and weight complete case analysis. If you decide for go for the conservative treatment, the available case analysis is just, again, just working with your data, forget about the missing data, and let's see how it goes. So, but there's another option which is less worse than the available case analysis, and it's just try to weigh the complete case analysis. By which I mean that you will need to, for those variables that, or for those patients that had all the variables, they are going to have more weight than for those variables that didn't have the complete data set. This is another strategy, but to be honest with you, none of them are recommended nowadays. And the reason for this is because we have options for the interventional treatment. So by interventional treatment, I mean basically three things. Something that is called a single imputation, and we are going to work with this data if we have time. The other one is called maximum likelihood estimation. For those who attended the July statistical workshop, I'm hoping that you might have an idea what the maximum likelihood is, but this is a really abstract concept, really difficult to explain. And you have to understand the theory behind the logistic regression, the regression models, before understanding the likelihood estimation. But I brought this just to mention it because that this would be perfectly another topic for another meeting. And the third interventional treatment would be the multiple imputation. So if you ask me what would be the best one, so the best one is definitely the maximum likelihood estimation. You ask me what is the most common one. The most common one is the single imputation, and the multiple imputation. There are several ways to approach this. We are just going to address some of them with the data. And because the single imputation is so common, so popular in clinical research, we have to mention this, but it doesn't mean that this is the best option for the treatment of your data. And the reason for this is because the single imputation is usually well accepted by journals, editors. So this is why you often see the single imputation in the literature. But again, it has a number of limitations, and you have the chance to go for the multiple imputation, go for it. I mean, for me, that it makes a lot of sense to go for the multiple imputation, and you will understand. Why am I saying this by seeing our exercise in this data. So single imputation. So basically, I know many of you won't understand what I mean by this, but I'm really hoping that when I'm explaining to you these concepts in the data, you will understand better this. But basically, when you are having missing data, you could replace just by the mean of your variable, provided that your variable has a normal distribution, or the median of your variable, provided that your variable has not non-Gaussian distribution. This is it. So I'm going to explain to you more in depth this with this data, but it's really simple, really. Hot and cold deck imputation. That means that whenever you read this, that means that the investigator chose match case, a very similar case, very similar patient, very similar participants, and just copy the missing data that they have, and that's it. So whole imputation is when you are matching for the data set of your trial or the same clinical trial, and cold deck imputation means that you are retrieving those data from other clinical trials. So you just need to find a sort of matched patient and try to replace those data. But again, it has a number of limitations because it doesn't necessarily mean that it would be equal for the other patient, even when the patient is matched. This is rather unusual to see the hot and cold imputation, and something that is more common is the last observation carry forward. So basically, to just replace whatever the data is, whatever the number is, by the last observation of your variable, which doesn't make sense to me because the last patient could be completely different from the patient you are trying to replace the missing data. But you have also to keep in mind the context of your clinical trial. You have to understand your data set to see exactly what would be the best strategy, because in some instances that would be a good idea, but it depends on your data set. It depends on your study as well. Okay, baseline observation carry forward. So again, it's very similar to the last observation carry forward. It's just that you go for the first one, for the first observation, and then you replace all this data with the baseline observation. It might make sense in terms of, for example, clinical trials looking at pain scores, for example. And so if you have a missing data, so and you go for the patient that had the worst pain score, so that would be a more conservative analysis. I think this is the worst case analysis. The worst case analysis, this is an example I would like to say in terms of the pain scores. So if you choose, let's imagine that you miss a pain score for a given patient, so you just gave 10/10 in the worst case, assuming that the patient had the worst pain and the medication didn't help. So this is the worst case analysis. That might make sense, but the problem with this is that you might actually be very conservative and increase the possibility of type 2 error and not finding any difference between treatments, because you had a lot of missing data, let's say 40%, and all of these 40%, let's imagine that you decide to replace the pain score by 10. So even when the treatment was effective, you won't have any difference. So this is type 2 error. I don't know if you got my point. And finally, there's something more interesting, which is the regression imputation. So basically, you just run a regression model, and based on the variables for that patient, for that particular patient, for example, the age, for example, the gender, the race, based on all these variables, so you can actually try to predict what would be the missing data for that specific patient. Just running a regression imputation. We are going to run a regression model in 10 minutes or so in our Stata. And finally, the multiple imputation, which is a set of linear regression models. I will definitely explain to you more in depth this, because I think this is the main reason we are running this data today, to explain to you the multiple imputation, because all of these are are easier to understand. The mean, the hot and cold, the lesson. The multiple imputation might not be really straightforward, but hopefully with that example in this data, we can understand better what we mean by multiple imputation. Any questions so far?
Wilson, I guess it's important to say that the assumption for all these intervention treatments is that the missing data was at random. And that's the why I guess you started saying that not completely at random, there is nothing to do. Your result will be biased. So prevention is a fundamental strategy to have. But when you plan this missing data strategy, you must keep in mind that your main assumption is that the missing was at random.
Yes, definitely. Yes. So this is, unfortunately, this is something that we can't really prove, but this is an assumption for all of this. This is a single imputation, maximum likelihood estimation, multiple imputation are assume that your missing data set was caused by the missing at random mechanism. But as I say, there's really no way to demonstrate this. Okay, so follow up. How do you once the treatment is done, you have to do a follow up. So the follow up is just the sensitivity analysis. So just to make sure that the data you replace makes sense. Yeah, and rather than making sense, your data is consistent with the values that your Stata software gave to you. I mean that you're not, for example, if you are trying to replace the missing data of body BMI, and the BMI Stata is giving you a BMI of 50 or 60, so it doesn't really make sense. So you have to look at your data and do a sensitivity analysis to see if the changes are consistent with your replacement, with the data that Stata replaced for you. Okay. So the prognosis again, the prognosis of the missing data again, it depends on the mechanism. I already explained to you that the missing not on random has no solution. Unfortunately, you will have biased results. The missing completely at random has a good prognosis, and the missing at random has a good prognosis provided that you have sort of treatment for this. Okay. And obviously the aftermath of the sequels are basically the bias estimates, the waste of resource, the waste of money, frustration, because you couldn't really find anything and your results are completely biased. Okay. So this is another way to explain to you the mechanism of missing data. In this case, is the missing completely at random, as Ana Maria explained to us very well. So in this case, just imagine that the top charts are before starting the trial, and the bottom ones are after finishing the trial. So you will have a proportion of missing data. For example, from here, 150 patients, male patients started the trial, but unfortunately 50% withdraw the trial. Same here, 50% withdraw the trial, which is quite a lot. But what I would like to show you with this is missing completely at random, because it has nothing to do with the outcome. And it has nothing to do with the outcome, because you can see here 150 dropped to 75, exactly the 50%. But these ones from 50 dropped to 25%, so 50% again. So it has nothing to do with the outcome. And you see the variable, which is the gender, which is the observed variable. So you will see that in the case of women, 60 women start the trial and 50% drop the trial, 140 and 70. So it has nothing to do with the outcome, because in both sides they are dropping exactly the same. This is just a theoretical example for you guys to understand better this concept. And on the other hand, it has nothing to do with the observed variable, because the observed variable in this case, which is the gender, both of them dropped 50%, but just imagine that women dropped more than men. So this is another mechanism, but we are talking about missing completely at random. So this is what I mean by missing completely at random. Has nothing to do with the outcome, has nothing to do with the gender, and therefore the risk ratio was 1.5, 1.5, and 1.5 here, when you sum up the men and women. Okay, so this is missing completely at random. On the other hand, missing at random mechanism, so is exactly the same. Here, they are dropping exactly the same proportion here when the outcome was positive and when the outcome was negative. So it has nothing to do with the outcome, really. This is why it's called missing at random mechanism. Because if the missing data is related to the outcome, this is immediately missing not at random, missing not at random. Okay, so in this case, because it has nothing to do with the outcome, this is a MAR mechanism, and the difference is that the proportion of men that drop the trial is different from the woman, so it has something to do with the gender. For example, 150 here and drop to 120. But the proportion is different here. For example, here it's from 60 to 30. This is 50%, and from 150 to 120, this is not the 50%. So there's a considerable difference between gender, and this is why it's called missing at random. So as you can see here, before starting the trial, the pool estimate for the risk ratio was 1.5. And because of this, because more, in this case, more women withdraw the trial, so the risk ratio is going to be slightly different. So this is just a theoretical example of what would be the MAR mechanism, okay? And finally, the missing out at random mechanism. So again, it has to do with outcome. So in this case, for example, here, 80 patients started the trial, and then 70 finishing the trial. So only 10 of them dropped when the outcome was positive. And when the outcome was negative, 120 and 60. So you see the difference. Here, when the outcome was negative, was 50% of the dropouts, and here just 10 patients withdraw. So something happened here with the outcome, and this is why it's called missing not at random. Okay, I hope this is more clear now. And this is the relation imputation. So again, it might be more, it might be easier for you if you attended the July statistical workshop. But basically, so you got, in this case, data set, a very small data set of, I don't know, I think there are 15 patients here. I mean, this is a very, very small data set, but just to show you what the regression imputation would look like. So every point is a patient, okay? So you just construct your regression model, and based on your line of the linear regression, this is a linear regression model, so you can actually predict what would be the missing data for that particular patient. For example, if this patient had an X value or something here, so the the missing data will lie here. So you just need to rely on your regression model, on your linear regression model. Again, I will give you some examples for you guys to understand better this. And multiple imputation is exactly the same, but let's imagine that you've got, in this case, you've got, for example, say 100 patients or even 200 patients. And then you are, your data is going to choose randomly. In this case, there are 15 patients. So let's imagine that from those 100 patients, your data is going to choose randomly 15 patients, and then this data is going to construct a linear regression model for you. Okay, so in a second step, Stata is going to choose again randomly another 15 patients, and based on these observations, these red points, Stata is going to construct a second linear model regression for you. And this is going to be an iterative process. So another 15 randomly chosen patients are going to be here represented by the yellow dots. And then a third linear regression is going to be there. Same for the gray points and same for the blue points. So in the end of the day, what Stata is going to do is just to merge all these linear regression models, and then is going to pull the estimate for you, and it's going to provide a confidence interval, which is represented by the green shape here. Okay, so this is the multiple imputation. So you've got 5 data sets chosen randomly from your original data set, 5 different linear regression models. And once you've done this, so you just merge all this information to construct a single linear regression model, which is going to be eventually the base for your prediction. This is more accurate, more consistent, more reliable, because it's based on, rather than just one linear regression model, Stata is going to construct a number of regression models. But how many regressions or how many data sets are recommended? So it depends on your proportion of missing data. If the proportion is really high, so it's usually recommended to have more data sets. But generally speaking, with five of these, 5 data sets is enough if you have, let's say, 20% of missing data. But if you have more, you could go up to 20. So between 5 to 20, you will be fine. Because obviously when you are doing this with this data, you have to tell Stata how many data sets do you want. So Stata it will do whatever you want. So if you say 100 data sets chosen randomly from your data set, Stata is going to do 100. But just keep in mind that you say 100, Stata is going to take a long time to construct this information for you. Okay, so again, basically, generally speaking, it should be between 5 and 20 data sets. Okay. So we finished with the first part. I don't know if you have questions. Otherwise, I will go straight to my example in Stata.
I had a question, Wilson.
Yeah.
Hi. When your missing data is not only missing at random, but also include missing not at random, what do you do in that case?
So you have to treat it separately, but the problem is that you can't really tell. I mean, you can tell, as I said, you can't really say, oh, this missing data is because of missing at random, and this is because of missing not at random. This is more, I mean, to me, this is more theoretical, concept, because in real life, it's very difficult to make a difference between those mechanisms. But if you manage to do so you should just treat it differently. Because if you have one variable with missing not at random.
Sí, ¿no? Y sí, bueno, no las agarres, pero bueno, mañana trata de pasar. El tema es que, claro, me piden...
Florencia. So yeah, so basically, I don't know who was speaking before. Was it Karen?
Karen.
Karen, yeah. So yeah, basically, if you manage to do so, if you manage to make a difference between them, you should treat them accordingly. If it's missing completely at random, so you shouldn't do anything. And if it's missing at random, so you should go for the treatment. That's I would say.
So if I'm not going to be able to really know the difference between missing at random and missing not at random, should I, in the cases that I have that doubt, treat the data missing at random and write later that we're not sure about this, but we did this?
Yes. So as I explained before, I think it's really important to look at your data set and try to look at some patterns that would give you an idea of what happened with your data set. You are really concerned. Yes, Karla.
I guess, Wilson, I don't know what you think, but maybe sensitivity analysis in this case could help. And maybe if you guess you could have not at random missing data, you can write as a limitation. For example, I guess I had 10% or I don't know, 2% of missing data because of severity of the disease, and the placebo group, we have a higher rate of missing data. So I think it's much more than only treating the data, but also including this in the discussion, the limitation of the trial.
Oh, definitely, Karla. Yes. So you have to try to guess a little bit what happened with your missing data, especially when you had lots of missing data. I think the discussion section is the right place to try to make hypothesis of what would happen to your data set. What do you think happened? Why your patients drop a lot? So yeah, and regarding the sensitivity analysis, I think it's a great idea as well to compare what would happen with and without the treatment. So that's a good alternative as well. Is that helpful, Karen?
Yes, thank
Unfortunately, there's no answer to your question, basically, because I mean, we wish we had some means to identify what MAR is, what MNAR is, but we don't have, definitely we don't. So if you go for the treatment, you have to assume that this is MAR, but the sensitivity analysis is a nice option. Okay, great. So so the second part of this meeting, what I would like to suggest to you, let me see.
Wilson, Emma asked if we, for the imputation model, the Stata will use the source of the data available in the study. And I guess, yes, the statistical test will use the data available. So it's a tricky point also, because if you had a huge missing data, it will influence even using the more fancy and the more sophisticated method to imputate your data, but to treat the missing data. But yes, they use only the available data. And also Saline asked the role of sensitivity analysis in missing data. and I guess it's great to discuss also. And then Danitza asked the model congeniality. It's a multiple imputation method that use another strategy than linear regression. Do you know, Wilson? I never heard about it.
I'm not sharing the screen anymore, am I?
No.
No. Okay. So the problem with the missing data is that this is such a complex topic. This is more complex than you think. There are books about missing data. I'm just explaining to you the basics of this. Is what I just explained to you is the basics of the missing data, but it doesn't mean that this is the only available. And this is, on the other hand, this is an evolving discipline. So people are trying to develop more and more strategies to treat the missing data. I haven't heard about this. There are, for sure, there are more alternatives apart from the multiple input, not the multiple imputation, but the regression imputation. For sure, there are more alternatives, but first, we can't really cover in this course, and second, we don't have that knowledge to explain to you all this complexity. This is just a really basic introduction for what the missing data is, what the basic missing data means in your clinical trial. Yeah. Great. So
I have one question, one final question, Wilson. What influence does or may have the distribution of the available data? So it means the normal distribution or not normal. That means if the missing data, let's say, are 40%, and the available data, the resting data, have, let's say, a normal distribution, I can imagine, I don't know, that this may have an influence on, let's say, on the, assumption of MAR, for example, but also on the handling methods of the missing data.
Not that I'm aware of. From what I've read so far regarding missing data, I haven't encountered any contraindication to run, for example, a multiple imputation in case of non-normal distributed data. So I think the most important one is the assumption of the MAR, which again we can prove. But as far as I understand, there's no limitation in terms of the imputation. You asked me regarding the regression, so we have to go and revisit the assumptions of linear regression. Remember that one of the assumptions is the normality of the residuals, as I explained in the other meeting. But before running a regression, I assume that we have to do those sort of we have to prove those assumptions, especially in terms of the residuals. But yeah, in terms of the non-normal distribution, I don't think there is a limitation as far as I'm aware of. So great. So let me just share again my screen. So I would like to bring this example. Can you see this? Yes. I'm assuming you can see. Yeah.
Yes.
Perfect. So this is just an example that I found from the literature. This is a clinical trial recently published. It was published maybe a couple of months ago or something like that. And this clinical trial is looking at the effectivity of rivaroxaban or enoxaparin in non-major orthopedic surgery. So basically, rivaroxaban or enoxaparin are anticoagulants, both of them. So they just decide to run a clinical trial, which, by the way, was a huge clinical trial. And you can see here they involved 3,600 patients, and there went randomization. So it was a multi-center non-inferiority trial, double-blind, designed, randomized with parallel groups. It's a massive trial. So I thought that might be interesting to look to use this example for my data set in this data. So if you look at the statistical analysis, let me go for the statistical analysis. So they said, okay, so in the primary intention to treat analysis, we use multiple imputation to account for missing data as described in the supplementary appendix. So that means that they, as you can see here, to determine the non-inferiority of the rivaroxaban to enoxaparin, the primary analysis was performed in the intention to treat population, which means that all the patients who underwent randomization, and in the per protocol population. And the reason for the per protocol is because of the non-inferiority design. And you will see in a couple of weeks' time, this is an indication, the non-inferiority design is an indication for the per protocol analysis. But when you are running a non-inferiority trial, you have to do both of them. You have to do the intention to treat analysis and the per protocol analysis. And the reason for this, you will see in a couple of weeks again, but the reason for this is because the null hypothesis of non-inferiority trials is that the treatment is actually inferior, and the alternative hypothesis, that is the treatment that you are trying to test, is non-inferior. So in that case, the null hypothesis and the alternative hypothesis and all the way around, compared with the superiority design. You will understand this better in a couple of weeks. But this is why they chose to use intention to treat analysis and per protocol analysis. But then they said, again, we use multiple imputation to account for missing data as described in the supplementary appendix. So I went to the supplementary appendix, which is here. This is the supplementary appendix provided by the authors. And if I scroll down to the page, I think it was the page 11. Okay, so the protocol specified that no imputation, again, that no imputation will be performed to account for missing data, which I think it wasn't really appropriate, because they were recruiting 3,600 people, so you have to have a plan for this. In my opinion, I think this is wrong a little bit, I think, because the protocol specified that no imputation would be performed to account for missing data. So they didn't plan. They didn't have any plan for missing data. And they run a very huge study. So if you're you are recruiting 3,600 patients, you definitely need to have a plan for your missing data. You will have missing data for sure, okay? So, however, given the substantial proportion of non-evaluable patients, multiple imputation was performed. So this is why what I wanted to show you. A logistic regression model was then used to estimate the risk ratio and corresponding 95 to side confidence interval, and a total of 100 complete data sets were generated. Okay, so remember when I said that as a general rule, we should use between 5 and 20 data sets, but because this was a huge clinical trial, so 100 data sets is appropriate, I think. And with treatment effect estimates combined across the data set using Rubin's rules. So Rubin's rules is basically this is a methodology to pull all the information coming from the multiple data set in a single number, and in that way to provide the imputation for that particular patient. Okay. And a sensitivity analysis was performed in which the rate observed in the enoxaparin group was applied to patients with missing information, the rivaroxaban group, and a rate of 0 was applied in the enoxaparin group. Okay, so this is the worst case analysis. The worst case analysis, the single imputation, and so it's interesting. I think that for me it was interesting this trial, that this is why I decided to bring to the discussion. Okay, so based on this, in terms for the sake of time, I'm just going to go straight to the data. So I'll just prepare something for you beforehand. So I'm just going to use an imaginary data based on this example. So I have here 30 patients. So let's imagine that instead of 3,600 patients, there were only 30 patients. I'm not going to do the 3,600, obviously, but I just made out some numbers here in terms of the age, in terms of the gender. So you imagine that 1 is for male and 2 for female. In terms of the treatment group, so these 15 patients, let's imagine that these 15 patients were assigned to the rivaroxaban group, and these other patients were assigned to the enoxaparin group. And then I have created another variable. Yes.
Excuse me, but your Stata screen is not sharing. I'm not sure if you are on Stata, but your screen is actually frozen.
Oh, sorry. Okay.
I thought it was only in my screen. Sorry, guys.
But did you see the paper?
The paper,
yes. Yes,
Oh.
we did see, but now it's kind of frozen.
Let me see what happened here. Share the screen. Share screen and share.
Yes, it's okay now.
Oh, okay. Sorry about this. So I was mentioning that based and inspired of this on this study, I decided to just make up some numbers here. Obviously, this is just an imaginary trial. Again, I decided just to include 30 patients instead of 3,600, obviously. So you got here the age of the patients, the gender, 1 for male, 2 for female, treatment group. So you got 15 patients here assigned for the rivaroxaban.
We are seeing the main screen of Stata. We are not seeing the data editor.
Group, and other 15 patients for the enoxaparin group. And ethnic, let's say there are 4 categories, whatever they are. Doesn't really matter, but they are 4. It's a categorical variable with 4 levels.
Wilson, sorry, we are not seeing the data browser page. We are only seeing the main page of Stata.
Oh, how strange!
Yeah,
okay.
you need to change your screen in the green button. So yes.
Oh, okay.
Now we are. Thank you.
Okay, so let's go for the third attempt. Third attempt. Great. So again, can you see this?
Yes.
Yes, great. So I was mentioning 30 patients here. There's a lot of variables. Just imagine this trial, right? I'm just trying to make up some numbers in spite of this trial, this clinical trial. So imagine that the age of the 30 patients was there here, and the gender, 1 for male, 2 for female. Number 1, this is the treatment group, by the way. So number 1 for rivaroxaban, number 2 for enoxaparin. Ethnic, so I decided to go for 4 categories. This is a categorical variable, so whatever the race is, doesn't really matter. And sorry, I didn't tell you that the primary outcome was a composite outcome in that trial. So this is the primary outcome of the trial, which is major venous thrombosis. Is a composite outcome that they use, and they use dichotomous primary outcome. So in which case MBT is 1 or 2. But now that let me just do something very quickly, because I'm going to run a logistic regression model. And in order to do a logistic regression model, you have to change the code of your primary outcome, because otherwise it's going to it's not going to work, the logistic regression. So so the primary outcome should be 0 or 1. So this should be 0. Oh. Sorry, let me just do this very quickly. 1, 1, 0. This is 1, 1, 1. This is 0, 1, 0. Oh, so by the way, this is really important because if you don't codify the primary outcome as this, and you go for 1 and 2, you can't really run a logistic regression model, as you might already learn in your statistical course from July. Anyway, so this is the primary outcome. The length of the operation, they include the length of the operation as well. So let's imagine that the length of the operation is this one in minutes. So this is a continuous variable. Sorry for the misspelling of the length. This is the BMI of the patient. So again, here, and then I just decide to just imagine that this is the complete case analysis. I mean, imagine that this is the ideal clinical trial with no missing data. Okay, so what I'm going to do now is just assume that I didn't get this number. I didn't get this from the BMI because we've lost the patient, we've done the follow-up or something happened. Same here. Same here, same here. So basically, this would be another variable, which is BMI missing. And this would be actually my actual data, okay? So let's imagine that I run a clinical trial with 30 patients, and unfortunately, 1, 2, 3, 4, 5, 6, 7, and 8 patients withdraw or lost the follow-up, or something happened with the measurement of the BMI. So I couldn't really measure the BMI, okay? So what am I going to do then? So, so this is the baseline observation carry forward. This is what I explained to you before. So in this case, the baseline observation would be the less number. Let me see here, for example. I think the less number was here, 19. From this example, so let's go with this for 19. So just replacing this for 19, okay? Because why? Because this is the baseline observation carry forward. So let's say this is 19. So I don't know if you understand this. I mean, baseline observation carry forward, just as simple as this. You just go for the baseline observation of the BMI. But as you can see here, my replacement here was 19, but the actual number was 23. Here 19, but the actual number was again 23. Here was 19, but the actual number was 27. So it's not really, again, it depends on what you're talking about. In this case, it doesn't make any sense, as you can see. I mean, there's absolutely no correlation between 19 and 32 or 19 and 34. So this is a very bad way to do the missing data in this particular case. In other data set might make more sense, but not in this case, as you can see here. So the other possibility is the last observation carry forward. So basically, so you just replace this number by the last one. So the last one was 24, that's why it's 24. And then the last one is 31, so 31 here. The last one was 26, so 26 here, and so on. And this is the last observation carry forward. Again, it doesn't make too much sense to me, but the last observation carry forward is very popular and is very well accepted by journal editors, and this is why it's very common. It's very often used. But I mean, you see that there is actually no correlation between, for example, this 23 and this 24, for example, or this 31 and this 23. So this is a very bad way to replace the missing data in this particular data set. If you give me another data set, I would say, oh, it makes more sense, really. Okay. So the mean imputation again, yes.
Student has a doubt regarding the baseline observational carry forward, because you added 19 in all the missing data, and they thought that it's the baseline for each participant. For example, if the baseline of BMI of participant 2 was 20, the baseline, the BMI baseline was 20, you should add 20 and not, for example, it is planned to be for each participant a different baseline. Okay, everyone could have the same BMI, but it's not expected to all missing data has the same.
Yes, exactly. It doesn't make sense in my data set because I didn't measure before and after, right? So this is why you shouldn't really use the baseline observation carry forward, because it basically makes no sense. But I mean, obviously,
but why you choose 19? I guess students, and I didn't understand why you choose the value 19. It's just an example, or
it's just an example because the baseline of the BMI was 19 here. But again, I'm not saying that it makes sense. But if you have a different data set, for example, you are measuring whatever the measuring is now in in a week time, in 2 weeks time, and 3 weeks time, and so on. So the baseline observation is the baseline that you had now, before, for example, before starting the intervention. In this case, it makes more sense. But again, what is the problem of the baseline observation carry forward? That you have too many cells to fill out, so you will have lots of data equal to the baseline, which means that there was no effectivity of the treatment, because it didn't change. It didn't change the from the baseline, because you're including the same baseline. And the rationale behind that is because it's more conservative. And it's more conservative because it increases the type 2 error. The more you have missing data, the more baseline observation are you going to have, and then the less possibilities that finding a difference you're going to have. This is an indication of, this is not an indication of baseline observation carry forward. As I said, I have to say this because I didn't measure the BMI before, after, or whatever, okay? So, but it's just an example, which doesn't make sense in my data set, but it shouldn't really make sense in many instances, unless you are measuring the basal, the actual basal of the patient. Okay, so I hope this is more clear. So the last observation carried forward, as I said, this again, it doesn't make sense in this case, but if you are measuring, let's say you are doing multiple measurements, it makes more sense. For example, you are doing the measurement now, and in 2 hours time, in 4 hours, and 6 hours. So in this case, if you are missing the 6 hours measurement, you could replace this with the 0 hours or with the 4 hours. So if you replace with the 0 hours, is going to be the baseline observation carried forward, or if you replace with the last observation, which was the last you had in that particular patient, this is going to be the last observation carried forward. But because in my example, I didn't really measure, they didn't measure the BMI multiple times, so it makes no sense, but you are right. I mean, you should have the information from the same patient, really, because otherwise it doesn't make any sense, as in this case. It doesn't make any sense at all, okay? Is it clear?
Yes, thank you.
Great. So the mean imputation, again, is the mean of the of the multiple variables, which in this case is not appropriate because I didn't measure multiple BMI. So, but just it was just to bring the examples here, okay? And then same for the median imputation when you are having you're missing data with non-normal distribution. But again, I think you got the concept, because this is actually very easy to understand, these 4 concepts: the baseline observation, the last observation, and mean and median imputation. But then the main reason to run my Stata was the regression model and the multiple imputation. Okay, so let's do. What can I do now? Let's try to do a regression model first. I'm not going to do all the steps for the regression model. I'm not going to verify my assumptions because it would take me a long time. Can you still, oh, sorry. Let me see what happened here. Great. Can you see this?
Yes.
Yes, we can.
Great. So, so you got my variables here. So you just need to run a regression model. So rec here, and as I say, I'm going to skip all the steps that you have to do before running a regression model. But remember that before running a regression model, so you have to consider what the outcome variable is. So in this case, the outcome variable for this trial was MBT, as I explained to you. Okay. So, but in this case, I'm not interested in the MBT. I'm interested in the BMI miss. So this is why this is the outcome variable in my model, which doesn't mean that this is the outcome variable in my clinical trial. Okay. So BMI miss, and then this is also important to be mindful to include your variables, because you can't really include these variables for whatever reason. You have to have reasons to think that these variables, for example, might or might not be related to the missing data. So this is also important as well. Okay, so let's imagine that you decide to include as independent variables the age, the gender, the ethnic, and the length, for example. So just run a regression model, and then you will have the coefficients here. So you, again, we explain this outcome in our linear regression lecture, but basically what you need to do is just replace the data with every single patient with these coefficients. For example, I'm just going to do for the first patient. Let's imagine that this first patient had some missing data here. So can you still see here? Yes, I think you can see. Can you see my screen?
Yes.
Yes, perfect. So so I'm just trying to bring give me just 1 second. I'm just trying to bring the example for the first patient. So for the first patient, the age was 23, the gender was male, the treatment group was, in this case, was, for example, rivaroxaban. The ethnic was, let's say, white. The MBT was 0, which means that he didn't have the outcome, and the length of the operation was 34 minutes. Okay. So for this particular patient, and remember that in order to try to predict the VMI miss, you have to start with your alpha value, which is this one. So that would be for this patient, for the first patient would be 23.26 plus the age which was 23, so 23 times minus 0.04 plus the gender which was 1. That means that the gender, in this case, the gender was male. So this one multiplied by 0.47 plus the ethnic, sorry, because I didn't include the treatment group here, plus the ethnic was 1. So 1 multiplied by the coefficient of the ethnic, which was 0.033, plus the length. The length is here, so 34 multiplied by 0.101. So when you do this, you will come up with a number, which is going to be the BMI for this patient. I don't know if that makes sense. And this is only for the first patient. So you have to do for every single patient. And as far as I know, there is no way to do this. I mean, if you have 20 missing data, you have to do this 20 times. So it's a little bit difficult to
And you have to do by hand for each patient?
I believe so. I don't have any, as far as I understand, I mean, because you are running a regression model and you've got your coefficients, but for these coefficients are the same for all the patients, but you have to multiply by the data of each patient, and there is no way for Stata to do this. So it's going to be really a very difficult task if you have.
Yeah, I'm asking that because we have this task on the assignment. I'm not sure if everybody read the assignment yet, but this is one of the tasks of the assignment.
Yes, this is one of the tasks, and
Hi, everyone. I'm Alma. Yes, actually, there is a way I can share the command with you later, but there's a way that you can do it. You have to create a new variable, and then you put the formula there, and then we'll make the calculation for each of the subjects that have the missing data. So you don't have to do it manually.
Oh, perfect.
I love you, Alma.
Sure, I will put it. I will put it there. I can share it with you. Don't worry.
Oh, thank you for
Alma.
that. Thank you for that. Thank you for the clarification. So I don't really have the command for Stata to do this, but I'm hoping that you understand what the rationale is behind the regression. And something important to keep in mind is that the outcome variable is not the MBT anymore. Your outcome variable is the BMI missing in this particular case. Okay. Great.
Yes, because in the real life you don't have the complete data. Yes. You don't have the BMI.
No. Yes.
Yeah, you have only the BMI with the missing data. So it was a doubt in the chat that student was thinking that maybe they would use BMI to have the perfect regression, but we are running the regression exactly because we have missing data.
Oh, definitely. So, because if we had this nice BMI, so there's no missing data. So that's the end of the lecture. So so this is the BMI that the theoretical one. That would be one. That would be the one. But this is the one that we had actually at the end of the trial. Okay, this is the one we had to work with. Okay. Great. So this is the regression imputation, and finally, I promise you to finish in 10 minutes time. So I'm going to explain to you the multiple imputation. So for the multiple imputation, actually it's a little bit trickier, but still I'm able to explain to you this. So the first step is to write the command, which is MI. So you have to, in the same way that you have to say to Stata that you are going to work with survival analysis, as you saw a few weeks ago. So this is the same example, but you have to say to Stata, I want to work with the multiple imputation now. So the command is MI, which is short for multiple imputation. And then you have to set your data. So multiple imputation set, and then I'm going to say white, which is white is the distribution of your data that you want to have for Stata. So you have to you may choose the long format or the wide format in the same way that you have to choose the format for repeat measurements ANOVA. that we saw a few weeks ago as well. But it is my personal preference to use the white, but you can use the long form as well. Okay. Anyway, so MI, which is multiple imputation, set and give me a wide format. Okay. So the second step would be to register the variable that you want to replace with missing data. Okay, so multiple imputation reg, in this case reg is not regression, reg is for registration, and imputation, register imputation, and then what is the variable that you're going to use. So in this case, I'm interested in the BMI miss. Okay, this is the design, the layout of the command. Okay, multiple imputation, regression, register, imputation, and then the name of the variable that you want to work with. Okay, so Stata is just doing this. Okay.
Wilson,
yes.
Wilson, we are not seeing your data browse now.
You're not seeing my data. No, I'm not showing. Can you see this?
I can see the Stata.
Only okay. Great, great. So I'm going to show you what happens here. What happened here? Can you see this?
No, we can't.
Okay.
It's because you have to change your screen.
Okay, so
click the button, the green button. Yeah.
This?
Better.
Great, thank you.
So basically, what Stata did for me was this. One is for missing data here, missing data, missing data, missing data, and 0 for those who didn't have any missing data. Okay, and then I need to change again my screen, which is a little bit inconvenient. Okay, can you see this? I'm just in the commands.
We can see.
You can see the command screen, right? So once you've done this, and then the next step would be to impute the your data. Okay, so mi impute, and then again reg for regression, and then again you have to remember that the regression model is going to be constructed based on the BMI miss. And then you just need to include the variables you are interested in: age, gender, ethnic, length, for example. I'm just giving you an example. And then just put a comma there, and then you need to say to Stata how many data sets do you want to generate. Okay, so again, the number of the data sets, it depends on how much missing data you have in the data set, but in this case I'm going to set up only 5. Okay, and then there's another comma which is rseed, which means random seed. This is not as important as the number of the data set, but you just need to type some numbers here, which basically the function of the random seed number is to give some consistency to your estimations in the way that you log out from Stata and then log in again with the same random seed number. In this case, I'm going to say 1, 2, 3, 4. So you are going to have the same estimators that you had before logging out from the Stata. As I say, this is not as important as defining the number the data sets, and you just need to click here, enter, and then you had impute, as Stata said here, you have impute four observations here. So let's have a look here what happened. Let me see what happened here. Can you see this? I'm just trying to show you the browser. Can you see the browser?
No, we can't.
Okay, great. So, browser. Now, can you see this?
The data editor we're watching right now.
So yes, so this is what Stata did for me. So Stata generated 5 different data sets randomly, 5 different data sets from those observations, from the existing observations. And for every single data set, Stata for the first data set say that the BMI would be 27. For the second patient was 21, 21, 27, and so on. But the difference with the regression is that Stata is going to estimate this 5 times, because I say that I just wanted 5 data sets. So it gives more consistency to your replacement, to your data set. And then in the end, you just need to estimate this, pull these results in a single number. But just remember that for this particular example, the authors of this clinical trial decide to generate 100 data sets. So you can imagine 100 data set for more than 300 patients. For a data set with more than 300 patients. So you can imagine how big is that data set. Is really massive to generate 100 data set, but they had to because they had missing data. So you can imagine how complex would be this. But this is basically what I just wanted to show you. And finally, so you just need to estimate with the command MI estimate. Just let me see if you can see here. Hopefully you can see here. So MI estimate and then colon logistic. I'm just going to do a logistic regression for this. And then you just estimate, in this case, because you're going to run a logistic regression model. So in this case, the outcome variable is not going to be the BMI miss anymore, but the MBT, okay? So MBT, right? Which is my original outcome, which is the outcome I'm interested in for running the logistic regression model. And then I'm just going to include my age, my gender. I'm going to include obviously my treatment group. I'm going to include my ethnic. I'm going to include my length of the operation in minutes. And then I'm going to use this one, which is the one I just generated for the multiple imputation. Okay. So this is the way you have to construct your logistic model regression with including already the BMI miss. Okay. So based on this 5 data set, Stata was able to construct coefficients for this. Obviously already imputing this. imputing these values here, okay? So the sensitivity analysis, one of the way to do this would be just repeat this command. Let me see. I'm just going to repeat if I can repeat this command, but not with the BMI miss, but with the, what was that? The, oh, sorry, with the original data that I don't have here in my data set, but I'm just trying to point out that the way to do a sensitivity analysis just would be to compare this with the BMI miss and compare against your data set without any missing data inputs, and then you will see what the difference is. But basically, this is the way to do this. As you can see, it's very complex, especially in large data sets. But this is what the multiple imputation looks like in this data. This is what the regression imputation looks like. And as I said, all the other concepts of basal observation, last observation, mean imputation, and median imputation are very easy to understand. But I just wanted to point out what would be the regression imputation and the multiple imputation in Stata. And I think this is all I wanted to share with you. So I hope it inspires you to study more about this very, very interesting topic. Sorry, I didn't have really the command to do this, to do the regression imputation with this data. Actually, when I was a student, I had to do this on hand, and I was really
Yes, I had the same issue when I was a student, but Alma just shared the command in the chat, so students now are much easier.
It's because Alma likes us better than she liked you.
Yeah.
No.
That's the reason.
Alma loves me too.
But still, I think it's interesting to do it on hand to see what the data is doing for us. But obviously, I'm not going to do for a data set
very interesting, extremely
3,600 patients. But anyway, this is all I wanted to share with you. I hope you enjoy our presentation. Thank you very much for joining us. And final questions, I'm very happy to take some final questions or final comments if you like. So thank you very much.
Thank you, Wilson. Another amazing lecture, especially because when I read multiple imputations, a kind of abstract, a lot of multiplications and formulas and so on. And when you put in Stata with a real, at least a real, not real data set, but expect to be a kind of a real data set, it makes much more sense. And I'm sure students like it a lot.
I'm really hoping that you learned something and then you understood more the concept of regression imputation and multiple imputation, but more importantly, the importance, the relevance of the prevention, the missing data, which is really I think is the main message from this lecture.
Yes. Sorry. I want to add, you both guys done an excellent job. Missing data is not an easy topic to us to review. So your slides were outstanding, very clear. And I also want to add that in order to prevent the missing data, it's very important to define in the protocol which are the variables that you are looking for, because at the end you will face that not all the variables that you try to consider for your study will be accessible. Okay, so I think that this is one of the most important topics, as you mentioned, the prevention of missing data, because not only in interventional trials, but also in observational trials, it's very complicated. In fact, I'm currently working with a project based on the problems of missing data in observational studies. And I think that it's important to, as a recommendation, try to follow the Equator Network guidelines, which specify very clearly about all the not as a quality, but as a guide to the recommendation of what to report. Because at the end, our studies and our results will be published, and not always the journals are following the guidelines from the International Committee of Editors, which is a real problem. So that's what I want to add. Once again, excellent job, Wilson. Thank you for showing us the Stata platform, which is also very complicated sometimes. So I think you've done an excellent job, and your slides are great. Thank you also to Karla for being present in the chat and trying to answer all the questions from our students. I think, guys, that you both done an excellent job. Thank you very much.
Thank you very much for your nice comments, and I'm really glad to know that it's being helpful for many people, and we will try to organize some more meetings for you very shortly, so we'll keep you updated.
Yeah, so great, Wilson and Karla. We appreciate a lot all your effort. Wilson, your slides, I like all the way how you present the data and the explanation to the students. All that creation that you do for every lecture is amazing. And everyone else, how we're talking about that. They saw that there was from some other presentation, but it's actually your own work. And I and all of us in the program and the PPCR team are really proud to have you both doing these lectures. And don't worry about the command. I just know it because I like Stata, and I generate command, and it have a displayed commands of how to add the calculations easily. It's not that you have to do something special for regression. So it's just that I'm really sorry that you have to do it by hand, but I also recommend the students to do it because that's how you actually understand from where that new value is coming from. Okay, and remember that there are many other methods to do it. Of course, regression is really detailed and it's actually focused on each of the subjects, so it will help you a lot. But I also have to understand what is the reason behind the missing data, so that's really important. And Wilson and Karla, they did a wonderful work explaining that. And guys, we have to cut because Wilson is being waited in another program right now, and he's really late. And I really appreciate that you took your time for 40 more minutes here with us today. So thank you all, and we'll share the recording, and we'll see you again soon.
Nice to see you guys. Thank you very much. Thank you.
Thank you. Bye. Bye-bye. Great presentation. Good luck, Wilson.
Thank you. Thank you. Bye.
Wilson is amazing. And MTLI is waiting for him. So yes, I don't hear you. You're muted, Karla. But
yeah. Alma, Wilson is fantastic, and
both of you work really great.
Because he really loves the statistics and all this, so we can see this just watching him. So
great.
Thank you, Alma. Thank you so much.
Thanks to you guys. So I have to stop so we can stop broadcasting too. But I wish you a great night and thank you again. And I'm here. Let me get back to you on the sample size. We have to review that, but I have to right now stop broadcasting here because we're on live on YouTube in the PPCR program channel right now. But yeah, okay. Thank you again. I'll see you.
Sure.
Bye-bye.
Bye.